we'll dive into drug product here. But, April, during our pre-call, you were talking a little bit about some of the innovations you're seeing in terms of, the IVT technologies. What are some of these, you'd mentioned plate-based or bead-based IVT. What are some of the other, innovations, or walk us through where you're seeing some of these innovations playing an important role for us, and how prominent are they in this world today?
Yeah, and some of the ones that have caught my attention is, I think the concept of using beads for mRNA purification isn't new, but putting it all into a kit and automating it, just plug and play, is something Aventor's looking at, and I think that would be useful. I'm always thinking high throughput, and the easier we can make that, probably the better.
Yeah.
One of the interesting technologies is, and I think it came up on the poll as well, is these modular approaches to mRNA synthesis. So Nutcracker's doing this. I think there's probably someone else, I forget their name, but I think these have a lot of applicability for personalized cancer vaccines, rare diseases. So that's pretty cool. And I've looked at this technology, and the way it's set up, it's really interesting because it gets us away from having to have a whole clean room suite per product. It's a ballroom setup because it's very modular. So you can have, like, the facility's open, you control the cross-contamination by enclosing it in the module. And that reduces overheads and increases the amount of products you can handle in the facility dramatically. But scalability is going to be, so once you move out of the personalized cancer vaccine realm, and if you want to actually ship these out to folks, that they can just like plug in their units and make mRNA wherever, it needs to scale up for certain applications. So, so I like those, like, in concept. I just feel like when you're designing facilities, that's going to be much easier. And then there's innovations like Devron's now got the Codex high cap. It's the RNA polymerase. It reduces the amount of cap that you need, which is great because that's a key cost driver, and it reduces the double-stranded RNA. So we've been looking at that, and then everybody's got their cap analogs. So clean cap is great. There's more beyond clean cap. Everybody's making them now, so it's just great to see that healthy competition. And the aim is to reduce immunogenicity, but also improve the yield and the purity. So that's all great stuff.
Okay, you hit on a couple of really important points here. So one, I wanted to kind of take a pulse check, right, with Tyler and Adi as well, in terms of these modular manufacturing units, because this has started to create a little bit of, again, more waves. I think they were coming up more, the conversation at different conferences that I've attended in the past year. We're starting to talk more about this. And April had mentioned, this is going to be great for personalized cancer vaccines, right? I'm curious, Adi, maybe we start with you, and then Tyler, how do you see these technologies filling a particularly important need today for the mRNA space, and where are there still some gaps, right, that we need to consider as we're striving to make these approaches even more meaningful for the space?
Standardized modular manufacturing. I guess it goes very well with mRNA manufacturing, right? Because again, inherently mRNA manufacturing has a platformability associated with it. The only thing that you can, ideally the only thing that you should change is your template DNA, and then you should be able to get your product, whichever that is. So that helps, that inherent nature of this manufacturing step helps a lot. A few of these things April already mentioned. So, if personalized medicine is something that you want to go for, it needs to accommodate different constructs, right? So it's not something like a vaccine, which is meant for a large scale population where you can just make one particular thing at a really high scale. So rather, if you have an equipment that can somehow facilitate this approach of creating multiple RNA product from a very singular, I would say, plug and play approach. The main thing that you would be changing would be the input template DNA, and then you're getting the output in different RNAs, right? So I guess it goes with, like the fact that the audience think that's a relevant thing. I agree with it. I definitely agree with it, and yeah.
Tyler, what do you think? Are you seeing these meeting very important needs? Yeah.
It's absolutely an important need. I think, when we think about wanting to solve these issues of cancers in developing nations, where it can be very difficult. I think the logistics is very difficult though too. And I think I'd have to really sit there and think about, well, what would it mean if I had one site that could manufacture all these different neoantigen mRNAs and then distribute them? I think the beautiful thing about LNP and RNA is they can be lyophilized, especially for a vaccine, right? In a non-therapeutic relevant drug product. So my question too is, does it make more sense to have the modular approach at bedside versus having a main distributor that can do this, in another way? So I don't know. I think it's absolutely an approach we should look into more, and in maybe certain scenarios with certain types of, cancers or in certain, nations that might make a lot of sense. But I think that's something that we should really look into in the field and see which way, which avenue should we take for each of these.